So, so you, some of you will maybe recognize the soil analysis. So maybe start with that. So the soil reports, they come in all shapes and forms, right? So we have here one lab that does it this way. This is one from Australia. This is one from the US. This is another small one here in the lower mainland. This is one from Germany. So they come in all shapes and forms. One thing they have in common, so in the top they have the laboratory name, chain of custody number, report number, account number, client name and contact and so on. Then there are the actual soil test results, and then sometimes they give you, oftentimes for extra money, the soil fertility guidelines, so what you need to apply. For that, you need to know what crop and cultivar you have and the yield expectation and previous crops. Otherwise, those recommendations won't actually work. So when I look at the soil test report, and here again, one person or at least one person may recognize that soil test report from our trip in that Serena was talking about. The first thing I want to know is actually I look at the date. And the reason why I look at the date is the season. So if I see at the end of a rainy winter, I see low nitrogen or sulfur, I don't worry. On the other hand, if I see high values at the end of the season, that means, well, maybe I could have applied a little bit less. Another thing is I look at the depth. So most nutrients are in the first 15 centimeters. So if my test reports from further down, I shouldn't be too much worried if the results are in the low end, except maybe for those that are much more leachable, like nitrate or sulfate. They will, depending on the time of the year, you find them lower in the lower layers. And another thing is then the application. Is that fertilizer actually applied banded or broadcast? That's important too. It's also important for the way you sample. Now, another thing that is very important is pH. And Josh will later talk more in detail about pH, or maybe next week. But just so much that you often find two pH reported. And one is the water pH and the other one is the buffer. The water pH is that what you use when you put in your soil pH meter, if you have one, or your paper. That's what you basically see. Then the buffer pH, that has an importance when the lime requirements are determined. So when you have a low buffer pH, it means that you need a lot of lime. If it's high, well, you get away with much less. Ideally, you want to have, for most plants, somewhere between 6 and 7.2. For organic soils, it's much less. It's somewhere between 5 and 5.5. So in this case here, for example, except for the third sample, the one on the bottom, the pH is still good. But remember that if you start, when you apply fertilizer, your pH will go down. And that really, it goes down by something like, when you apply 100 pounds per acre, it goes down by about 0.1 unit if you apply urea, or about 0.2 units if you apply ammonium fertilizer. So sooner or later, those others, in a couple, let's say in 5 years or so, you would need to look into the others to correct that pH too. Another important thing that I look for is organic matter. It impacts so many different things, right? It has many benefits, but it's also impacting things like the release of nutrients. So in this case, the organic matter is not bad for a farm soil. I've seen much lower values, more in the valley, but it is really okay here. It is impacting the release of nutrients, especially sulfur and nitrogen. And it's also impacting the pH. So as I said, the more organic matter you have, the lower is actually your target pH. So at 15, if you have 15%, you probably are still good with a pH of 6. And another thing is that it also impacts the cation exchange capacity that I talked about in the previous presentation. But now don't get frustrated if you have a low organic matter content, because the organic matter content is a natural equilibrium in your soil. So if you have a soil that's naturally lower in organic matter, like sandy soils, then or in soils that are very warm where you lose a lot, or on the south slope as compared to the north slope, you have a lower organic matter just naturally. If you walked away, if you have a high organic matter content that you hold up artificially, and you walk away one day, it will come back to that equilibrium. And in fact, it's an uphill battle, and you will also lose a lot of nutrients because that's not the norm or the healthy level of organic matter. So we talked about the cation exchange capacity in the previous presentation, so the ability to retain nutrients. And as I said, it's dependent on soil texture, organic matter, but also pH. So at low pH, and some reports tell you that, that's the cation exchange capacity as you see it, and then the cation exchange capacity if you had a pH, a neutral pH like 7. And this is something you don't really, that you can impact or influence with the organic matter, not with the soil texture. That's nothing you can change. Mind, however, that the cation exchange capacity is relevant on the more recalcitrant long-term organic matter. So it will take a couple of years until you actually jazz up that cation exchange capacity. Fresh material won't do the trick right away. When you have a low cation exchange capacity, it means you have to split your nutrient application, because you're at risk of leaching that nutrients, right? So you have to be careful here. And soils tend to become acidic much more quickly as when you have a high cation exchange capacity. This is just an example. So when you have light colored sands, the cation exchange capacity is somewhere in the 3 to 5 centimole. That's the unit. Right? If you have more dark colored sands, then it's 10 to 20 and so on. Right? So one thing you need to watch out are units. Sometimes they're reported in parts per million, but if you want to know what that is in kilogram per hectare, then you multiply them 2 for the first 15 centimeter. If you sample down to 30 centimeter, you need to multiply that by 4, and you get kilogram per hectare. So if you are a pounds per acre person, just take off 10% from that value, and you got that in pounds per acre. Another thing is that milli-equivalent per 100 gram. Here, the international unit is actually centimole per charge, but it's the same numerical, the same value, so don't worry about that. That is the unit for cation exchange capacity. And then for the salinity, not reported here, oftentimes in North America uses millimoles per centimeter. The official unit is DC centimeter. DC Siemens per meter. Now, there is sometimes a tricky thing. Some labs report it in different units, and don't get a heart attack if sometimes you see something tenfold of what you expect. It can be just a different unit. And then nutrient recommendations. Sometimes the nutrient recommendations are given in phosphorus, just in P or in K, or sometimes as potash or as phosphate. And there are also ways of transforming one into the other. Now, confusing other methods. You sometimes see different types of analysis. You have here in this case bicarbonate and Bray. These are different ways of extracting your phosphorus, and that is also true for other nutrients, but it is more crucial now in phosphorus, as here the differences are sometimes huge. Some extractants extract more than others, right? And this gives you a different value. And just to make things more complicated, the difference is also confounded with things like pH and other factors. So make sure that you know what method has been used. And that is especially important when you look, for example, at conductivity. here it's not only the method. So typically the standard is what is called saturated paste. So the sample is saturated with water, and then the water is extracted, and this is a standard. Many recommendations work with that. You shouldn't have more than 1 or 2 dcsiemens per kilogram or millimoles per 100 gram. But the problem is that oftentimes it's measured in a one-to-one method. That means one mass equivalent of water with 1 of your soil, or sometimes even a 1-to-2 method where you have 2 times water compared to 1 time of your soil. And here you can reach actually those levels much earlier, right? But this is oftentimes not reported, and that depends really also on your soil texture. Now, the macronutrients versus micronutrients. So the macronutrients is nitrogen, phosphorus, potassium, but sometimes, and also sulfur, calcium, and magnesium. Most attention is on the first 3, so nitrogen, phosphorus, and potassium, and sometimes also calcium and magnesium. Micronutrients are making less than 0.1% of the total plant tissue. So what the physical and chemical characteristics of soil affect the availability and the uptake of those nutrients. For example, soils low in organic matter may have lower micronutrient availability. Soils that have very high levels of organic matter may actually account the same problem. Then soils with higher amounts of clay are less likely to be low in plant available micronutrients, while sandy soils sometimes encounter micronutrient problems. Soil temperature and moisture, we talked about that. They also make it sometimes more difficult for plants to get that to the nutrients. So what is now the adequate level? Is the higher the better? Well, maybe the only thing we can actually really say is something like, well, yes, there is a response to the plant. This is when you still see a growth increase, a yield increase. Then you have somewhere a maybe, and finally a no. So where you're sure there won't be a growth, a response, right? At the same time, notice that actually if you apply too much, it becomes toxic and your yield gets actually less. Your cost going up when you apply more than necessary, and then the environmental impact goes up as well. Besides, if one of those plant nutrients is a little bit deficient, you have problems actually. You can apply the others as much as you want. It sometimes doesn't really help. So what are the adequate levels? Laboratory often uses either book values, or they use what is called the percent base saturation. I would not rely too much on the percent base saturation. That has been, there's oftentimes no evidence. And what is also true, many of those values have not been calibrated for individual crop species or varieties, or there's very little data for BC, let alone for your area. So this, for example, relying on a base saturation that's from another soil report, you see here 4,470 ppm of calcium is considered as low at a pH of 8.3. Impossible to apply that much calcium to bring that in balance. So it's impracticable and oftentimes actually useless. So what are adequate levels? The soil test is the only indicator, not a crystal ball, right? So one thing you always need to check is the pH, right? Then observe your crop. Do you see yield or quality decline, right? And then, of course, the trend is always much more important than this individual soil test. So plan ahead and don't wait until you're at the bottom of the nutrients, because, well, you actually feed the soil, right? And then there are other reasons. You see that this is a deficient corn, right? A phosphorus deficient corn. But the problem is not, there's plenty of phosphorus in that soil, is that it can't breathe, right? Okay, for this soil, and that's my last slide. So what is the verdict for this soil test? Assumption, forage, 2 cuts. The pH for the third sample needs some correction. Nitrogen is low, but well, you know what? You have enough organic matter, and with some legumes, you may actually not need that much anymore. The phosphorus is low in the first sample, yes, but it will be provided in manure, and some will be provided by organic matter. But still, you need to include it in your fertilizer plan, and especially for the first sample, you need to probably include that as a fertilizer. Potassium is generally low, so that is definitely helpful here. And for the rest, I don't even have concerns. And even for the sulfur, which appears to be low, but sulfur is not really, that is another, let's say, a moving target. Relate that to some yield is oftentimes pretty difficult. So, and I think I'm a little bit over time, but yeah, thank you for your attention.